Today we start talking about the different aspects related to infections. We're going to talk about types of infections. We're going to talk about epidemiology. It's the piece that I consider clinically relevant. Okay, we're going to have interesting conversations, I hope, about this. So one thing that I want to mention is that not every microorganism that is known can cause disease. In fact, most of the microbes that we know about do not cause disease. They are not pathogenic to humans. One of the greatest examples that there are about 219, I think a little bit more than 200 different viruses that are pathogenic to humans. Total number of viruses that can be found in a single fruit fly bat is 58. The total number of viral species that is estimated on Earth, different estimates put it in from 2 million to 100 million different species. We talk about 200 virus species that are pathogenic to humans. Same goes for microbes. Same goes for bacteria and fungi, and most of them are not pathogenic to humans. Okay, it's number one. Number two, we carry a lot of microbes on the body surface. We call those communities microbiome or microbiota. Okay. So which sides of the human body, without looking at the table, which sides of the human body can you think of when we talk about microbiota? Skin. Huh? Mouth. Intestines. Nose. Which part? Upper respiratory. Ear? I'm asking, ear? Yeah. Well, external. Reproductive organs. Mucous membranes of reproductive organs. Now, if you think about this, upper respiratory tract, skin, we forgot eyes, by the way. Eyes, external ear, intestines, reproductive mucous membranes. What's common between them? Huh? Warm and damp. Skin is actually dry. Skin is really dry. Okay, so keep those in mind. Now, let's talk about sites. Can you think of something that is sterile in the human body? Stomach is, despite of the harsh environment, is not sterile. No. There's not a lot of microbes, but it's not sterile. Anything else? On the outside, they do make lysozyme, but they still not sterile. The surface of the eye is not sterile. Huh? Say again? When? When it's in the bladder, it's sterile. You're absolutely right. Not even about liquids. Like if you take human liver, cut it open, and take the tissue sample from the inside of the liver, is it going to be sterile? Yes. Well, normally. Okay. So what's the difference between, say, a human organ like a liver and places like skin or eye? Exactly. Even intestines, if you think about this. Yeah. Bingo. Now, one thing that often confuses lungs. Do lungs have a microbiome? It's a great question, because the general consensus so far, they're sterile. Even inside of the lung is sterile, because alveolar macrophages do a great job in completely cleaning it all the time. We do not have resident microbes. That makes sense? Now, what is the role of those microbes? They protect us, right? Protect by establishing competition with potential invaders. They compete for the resources, they occupy ecological niches, they simply occupy space, right? And that this is not it. They may play an important role in things like shaping your immune response, defining your metabolism, making nutrients, for God's sakes. Vitamin K is produced by the microbes in your intestine. I've already, I brought this up, the ability to digest seaweed is microbial, okay? I want to give you a few examples. Again, it's more of an FYI. Shaping the immune response. Well, before shaping the immune response, let's determine few words. So, microbes that normally inhabit various body sites, normally inhabit, are called resident. Okay? Things like Staphylococcus epidermidis on your skin, Staph epidermidis is the resident microbe. And when people say resident microbiome, they refer to microbial species that can be normally found in different parts of human body. Remember, we took samples from the mouth and from the nose, and we found in the mouth streptococci species. Streptococci of different sorts are resident microbes of the mouth. Okay? Different body sites will be inhabited by different microbes, and we will see that. So they vary, they differ from site to site. And as we grow, as we go through life, they may change. They may or may not be linked to age, but they definitely are linked to the lifestyle. Microbes are acquired by many ways. What is the first human exposure, usually, to the microbes? When is the first time when a person exposes him or herself to microbes? Yeah, when you're born. Where those microbes are coming from? Birth canal. Okay, that's number one. And then, babies crawl around, they lick the heck out of everything, and, then they eat food. Food is not sterile, so we acquire microbes from everything, starting from floor, ending with our relatives, mother and father and siblings. Now, speaking of the birth, if a baby is born via regular birth route, passes the birth canal, baby acquires microbes from the mother. It's going to be, first microbes are going to be lactobacilli. What if baby is born by C-section? What's going to be the first source of microbes? So who's going to be? Yes, it was shown that newborns that are delivered by C-section, the first exposure and the first microbes come not from the mother, but from the OBGYN doc that holds the baby first. Okay? When you have two people that move in together, I don't know, a boyfriend and a girlfriend, they move in together. Interestingly enough, in a span of several days, the microbiomes are largely shared. People exchange microbes extremely fast. My personal goal, maybe not this semester, but I really want to do the microbiome of the mouth of pets and pet owners. I bet there's going to be a lot of the same. Another interesting thing would be intestinal microbiome of pet and pet owners, but that's a little bit, higher on the list. You have to convince people to sample their own poop. It's kind of hard. Okay, now how microbiome can shape the immune system. Have I told you, because I have a little or short and long-term memory, did I tell you about hygiene hypothesis? Oh, it would be a great time to introduce that. What is hygiene hypothesis? Do we live in a clean environment? You think so? Go to Africa. Go to Africa. Go to Africa. You don't want to go to Africa? Well, if you go to places like Africa, South America, Southeast Asia, I'm talking about rural regions mostly. Hygiene is the word that people are not really familiar with there. Okay, so there are there's a continuous exposure to protozoa, bacteria, helminths all the time. Okay, now what do you do with fruits or vegetables before you eat them? You wash them, right? And believe me, they are washed. All fruits and vegetables that are important. I recently learned it. So for instance, produce items that are imported, they undergo certain types of treatment, which will retain the food value and the smell and the taste. What's this treatment is going to be? Oh, no, not pasteurized. Well, you cannot pasteurize mango. Yes, irradiation. It's gamma or UV, okay, so to destroy or reduce the number of microbes on the surface, okay? Now, let me tell you something. Mangoes are often infested with fruit flies, so to kill the fruit fly larva in the mango, they often gamma irradiate it, right? So there's a lot of treatment of foods. We eat pretty clean, we live clean, floors are clean, toilets are clean, we don't have like 6-foot hole that goes down or 2-foot hole that goes down. And in many places in rural areas of the third world countries, they don't even have holes at all. If, I don't know, adults would probably go behind the tree, kids don't even bother. Okay, they can do whatever they need out on the street. Okay, so it's different level of cleanliness. Now, all this long introduction was to introduce, bless you, the idea that being clean is not that good all the time. If you would compare the rates of autoimmune diseases, such as allergy, asthma, lupus, okay, between the developed countries, say United States, and developing countries, say, take any African country, what you're going to see is that while here, okay, I have asthma. My son has asthma. Okay, so I bet, I'm not asking you to disclose your medical information, but I bet there are people in this room who have allergies or asthma, okay? So autoimmune diseases. You go to Africa, there are none. You go to another clean country, there are plenty. You go to, well, no offense, less clean country, there are none. So it's a correlation between the exposure to microbes and reduced risk of autoimmune diseases. The idea, which is really hard to prove scientifically, is that when your immune system is being essentially trained when you grow up in a proper way, when it is exposed to microbes, it kind of gets diverted from your own tissues, if I may say so. When you do not have that exposure, development of the immune system is changed. Does that make sense? Okay? And You have a completely different situation. Your immune system develops in a way that it attacks your own tissues. It sounds very non-scientific, and it is, because it's hard to say. You have to take sets of identical twins, okay, and start, put one in the dirty conditions, put another in clean conditions, and see what's going to be the percentage of autoimmune diseases. Chances slim, right? I have the experiment in my own family, and I was blessed with twins. For every scientist, it's really a great achievement. I have one as a control, one as an experiment. One of them was treated with antibiotics when he was like 5 days old. He had an eye infection. It was pretty badass antibiotic treatment. He got tetracycline cream on his eye. Guess what? He's the guy who has asthma. He's the guy who has allergy. His brother, clean. Okay? I can see the difference. Of course, it's N of 1. So I do not have any statistics. It's anecdotal. But there are many anecdotal cases that kind of put it all together. So it's conceivable that our exposure to microbes shapes our immune response. Metabolism. Why did I say this? Oh, regulating metabolism. That's an exciting thing. Microbiome, human microbiome, is associated with obesity. It was demonstrated that in identical twins, one of them obese, one of them is not, they have different sets of microbes in the intestine. You put the obese, quote-unquote, microbes into the normal lean mouse. This lean mouse will become obese. You put lean microbes in the obese mouse, it will become lean again. I a little simplify the experimental setup, but the idea is that there is strong association between the intestinal microbes and obesity, intestinal microbes and diabetes. Okay? Whether there is some causation, obviously, because this microbiome transplantation definitely influences the body composition. The idea is that different microbial populations, they change how we extract energy from the food. Does that make sense what I'm trying to say? So I mean, if we extract more energy from the food, we're going to be obese. Okay, if we extract less, we're going to be less obese. Moreover, if you would change the diet, your microbial population will change. For instance, people who eat high carbohydrate, low fat diet, their microbiome, intestinal microbiome, consists mostly of the species, the class Firmicutes. However, if they change to high protein, high fat, low carbohydrate diet, they're going to have mostly bacteroidetes. Okay? Same is true if people start to eat high fiber diet instead of low fiber diet. How long does it take for microbiome to change? What do you think? Give me an answer. It's completely a guess question.
Probably within like a day or two if you
Day. 24 hours. So if you abruptly change the diet, like you decide from now on I'm going to eat 25 eggs a day, and that's it. Your microbiome will change overnight, 24 hours. That's pretty, for me, it's quite fascinating. It's been involved in so many things. There was a study showing the link in mice, of course, between what's considered to be autism in mice, they have a model, and microbiome. There was a study extending that to humans, showing that there's certain common pattern in kids with autism spectrum disorder and microbiome. So there is some crosstalk. Okay? Now, as you can imagine, resident microbiome always has guests. Those guests are called transient microbes. So they are passing through. Okay? These microbes can be pathogenic, can be not pathogenic. We, of course, mostly focused on the clinically relevant pathogenic microbial species. Okay? Now, some of the members, even members of microbiome, can cause infection sometimes. When, for instance, our defense is weakened. We have discussed the presence of Staph aureus. It's the component of the respiratory microbiome. If a patient becomes immunocompromised, the Staph aureus present in the throat or nasal microbiota can easily cause the upper respiratory tract infection. Microbes that use the opportunity to cause infection when your immune defenses are weakened are called opportunistic. Right? We good? Moving on. So this is the great picture which I provide for awe and fascination, mostly, with few points that I want to make. So this is the map of the human microbiome. It's a gigantic project. Human microbiome project that is led by the National Institutes of Health. It's very ambitious. What they do, they take samples of human microbiome from different sites and sequence the DNA of all the microbes that they can find there, and then determine, well, not complete sequence, but they determine which species are present at each body site. Some body sites represent an immense challenge to the scientist. Skin is easy, right? Mouth is easy, nose is easy, ear, eye, even eye, you can do eye. It's quite uncomfortable, but you can do that. Reproductive organs, easy. You know what is one of the hardest? Intestines. Because what we pass in the feces, we pass a lot of microbes in the feces, doesn't necessarily represent what's going on in the intestines per se. So now there's a new movement, kind of new idea, when people undergo colonoscopy. We're not talking about, taking biopsy without their consent, no, but to collect fragments of microbiome by washing the large intestine. Because, the endoscope is already there. You might as well just stick another tube and wash it a little bit, and you just need a couple of milliliters, and then you can throw it in the sequencing machine. So there's an idea to more precisely determine what's in the intestines. Now, these different colors here represent different body sites. So you can see stool in blue, cheek in yellow, so it's whatever's on the human cheek. Plaque on the tooth is red. Tongue is maroon. Green is nose. Purple vagina, and gray is skin. Now, there are some commonalities, and you can see that, for instance, there is a lot of commonalities between the cheek, tongue, and tooth, which makes perfect sense, right? But if you would look at the nasal microbiome, it's quite unique, for instance, right? If you would look at the skin microbiome, it is quite unique. If you would look at the intestinal microbiome, it's quite unique. So, for instance, not all the bacteria that you can find in a human mouth, well, most of them will be absent in a human digestive tract, and vice versa. So, I want to illustrate that different sites are occupied by different resident microbiomes. And our ability to determine which species represent microbiome are now so advanced that there is a branch of microbiological science quickly developing that is called forensic microbiology. Have you heard about it? So there are two approaches to do forensic microbiology. One is to trace down the bad guys. How would you do that? Well, say hotel room. I come into the hotel room. What I start to do immediately, unconsciously, start to shed my microbes all over the place. Okay? In fact, about an hour after I'm in the room, it's filled with my microbes all over the place. They're everywhere. Okay? Now next day, I leave the room. Before it gets cleaned, can you detect my microbiome? Not only you can detect it, you can trace it to me, not to any other person, because I have certain uniqueness to my microbiome. Does that make sense? What I'm trying to say is that we're all going to have streptococci in the mouth. The exact numbers,
It's like a fingerprint.
Yeah, it's like a fingerprint. Okay? It is not perfect yet, so it hasn't been used in criminal investigations, but people work on that. Another thing is corpses. So what happens to human after human dies? If you just leave the body somewhere, it decomposes. Now it's often an important task for criminal investigation to determine when the person was killed or when the person died, right? Now we have different medical criteria that allow us to determine with different degree of precision when the death occurred. Turns out microbes in the different sites of the body change in a very timed and precise way after the person dies. So there is, have you heard about Body Farm in Tennessee? No? There's a huge research facility, often referred to as Body Farm. People donate their bodies. When they die, their bodies are placed in different conditions. Forest, water, dry condition, wet condition, all kinds of different conditions. And scientists watch how the body decomposes. Of course, they look for classical presentations, classical signs, okay? But now there are studies of how microbiome changes after death. So you can later take samples of microbiome, and if you can find common patterns, it can help you, bless you, it can help you to determine, when the death happened. Does that make sense? Well, let's talk about types of infectious diseases. Some of these things are eye-openers. People have sterile sites in the body. What is sterile? Don't look at the table, it's boring. With the table, it's boring. What are the sterile sites in the human body? Inside of the organs, yes. You're like inside of the muscle, inside of the heart, parenchyma of the brain, parenchyma of the kidney, liver, spleen, whatever. CSF, very good. What else? Fluids. Which fluids are sterile? Blood is sterile. CSF is sterile. KJ mentioned urine before it comes out of the body. Sterile. Saliva? When it enters the mouth, yes. When it is produced in the salivary glands, it's 100% sterile. Same goes for tears. Okay? Even sweat in the sweat glands is sterile. Once it gets on the skin, that's a different story, but it acquires its microbes from the skin. Okay? Practically all the fluids are sterile. Right? So, now think about this. If you have a microbe of some sorts, okay. Salmonella, we just had Salmonella typhimurium right there. So if you put Salmonella on your skin, is it going to cause infection? No. Unless it's a wound. Bad wound. Okay? And we'll get back to the wound. If you put it in your intestine, is it going to cause infection? 100%. Or it depends on something. What if you put just a tiny little bit of Salmonella in your intestines? Depends on the infectious dose, right? Depends on how many cells you put inside. Okay? Now, if you put Salmonella in the blood, yeah, you're screwed. Why? Why it may or may not cause infection when it's in the intestines, but it will definitely cause massive. There you go. Sterile sites are at the highest risk, right? There's no competition. We talk about, and when we talk about wound infection, you essentially put the microbe in the site that is supposed to be sterile. Your tissue, your subcutaneous tissue, your muscle. We good in that? Okay. Now, when we say that someone is infected, what does that mean in relation to the pathogen? Pathogen must do what for the process to be called infection? Say second word? Grow. It must replicate, okay? Replication or growth. I can inject murine cytomegalovirus of the mouse in my muscle, and a couple of times I accidentally did that. It's not going to grow, it's not going to replicate in my tissue, so there's no infection. Does that make sense? Okay. Now, infection, although it may happen, does not have to have any symptoms. Such infection is called asymptomatic or inapparent. Did it ever happen to you when, I don't know, your family members all suffer from cold, for instance, and you're just fine? You're fine. Nothing happens to you. Does that mean that you're not infected? Not really. You still may have the pathogen. It still may replicate. Okay? But you just had no symptoms. Your immune system just shut it down completely. Okay, at certain points, so you didn't have symptoms. Against what? Okay. It's a great question. Technically, yes. There can be replication of the virus in your organism. The question is only, does it replicate to the levels sufficient to transmit? Does that make sense, what I just said? So people transmit infection because they produce whatever they transmit it with, say respiratory secretions. And those respiratory secretions contain a lot of virus. If you vaccinate it, you get virus in, it starts to replicate, your immune system shuts it down. The amount of virus in your respiratory secretions is extremely small. You does not transmit it to other people. Is there a chance you still will do it? Yes, there is. How huge this chance is? It's really tiny. Does that make sense? So in science, you rarely say, it doesn't happen. In science, we mostly say it shouldn't happen, or we reduce the probability of something to happen. Okay? Now, communicable diseases. I don't know how many M's I put in there. Communicable diseases. Can transmit from patient to patient. Can you give me an example or examples of communicable disease? Tuberculosis, great communicable disease from one person to another. HIV, well, there's a lot of clinically relevant stuff. Well, non-communicable disease cannot be transmitted from patient to patient. Can you? And we're talking about microbial, not cancer, not cardiovascular disease. Microbial, non-communicable disease. Yes, if it was auto-inoculation. Technically, there were cases when people shared the restroom and got it from each other. Infectious. I mean, it should be microbe. Non-communicable. Oh, they are, oh my ass, they are communicable. All the time. Otherwise, 1/3 of the world wouldn't be infected with parasites. You have very, very acute pain in the left lower abdominal quadrant. Sorry, not left, right lower abdominal quadrant. Appendicitis. And you say, I'll just pop up like 5 pills of ibuprofen and I'll be fine. It can happen to you in some time. Why? Huh? Okay, appendix will burst, and what happens next?
It's a whole host of stuff to do inside of the sterile environment.
So all the microbes?
You change that environment to a very productive growing environment.
Bingo. All the microbes from appendix get into the peritoneum and start to replicate. And they cause a hell of a problem. Is that an infectious disease? Sure it is. It's caused by microbe. There's an infection of your guts. Right? Well, peritoneum. All is infected. Just it's horrible. Can you transmit peritonitis, communicate peritonitis to another? No. You're going to die alone. Nobody else is going to suffer. Okay? That's an example of non-communicable disease. They're not as frequent. Okay? For instance, when we talk about non-communicable disease, if we talk about disease between people, when rabies get into the human rabies is non-communicable. We have no record of people biting other people. It's technically possible, but we don't have any record of it. Okay? Now, contagious disease. This is something interesting to discuss now. Contagious is sort of similar word to communicable, but we also sometimes use the term one disease is more contagious than another. Okay, which means more contagious disease is more easily transmitted from person to person. Is that clear? Disease can be more or less contagious. And the ability of people to transmit disease between them is measured, well, not measured, but characterized by the model of basic reproduction number, or R0. Okay? How it's calculated, I cannot tell you. I tried to figure it out and just couldn't First of all, it's a model. So it's not a measurable parameter, okay? It gives you an idea of how likely the person to transmit the disease to other people. Basically, on a very fundamental level, it shows to how many people an infected person can transmit the disease. Let's say you have influenza. Basic reproduction number is 2 or 3, from 2 to 3, which means one person with influenza infection is most likely to transmit influenza to 2 or 3 people during the course of disease. Does that make sense? That number is entirely model. What I mean by that, imagine, well, one of the great things too, why I love this table, and I'll be blunt with you, this is from Wikipedia. I didn't create anything, I just took it as an example. Remember the scare when there was an Ebola epidemic? So for Ebola virus, basic reproduction number is between 1.5 and 2.5. Say it's 2, okay? Which means one patient with Ebola virus infection is likely to transmit Ebola to 2 other patients. To give an idea how infectious Ebola is, if I would have an active Ebola infection right now, and I would be lecturing here, none of you would acquire the disease. It is not transmitted by respiratory route. On the other hand, if I would, now I know it's going to sound extremely repulsive, but if I would tie all of you to the chairs and will start to spit in your mouth, I would infect 100% of people in this room. Does that make sense? So this number can be applied to somewhat of a hypothetical ideal situation, normal social environment. One of the reasons why Ebola virus epidemic in Western Africa spread like a wildfire was social customs. One of the social customs that infuriated medical workers was when somebody died and there were proper funerals, okay, the custom is that every person who attends funerals have to come to a dead body and kiss him or her. Well, if the person died from Ebola, it's probably the easiest way to spread it to, I don't know, like 100 attendees, okay? So you have to understand that there are different conditions that would influence that number. But what is interesting about this table is that it maybe challenges some of the stereotypes that we have about infections. Things like Ebola and influenza are really not that infectious. Okay? HIV probably depends on the behavior. So if somebody exposes himself or herself to sexual contacts or leads more promiscuous sexual life than others, they're at the high risk. Also, it depends on the population, exposure type, okay? So one of the breeding grounds for HIV vaccine studies is Thailand. One of the main industries in Thailand is what? Sex tourism, prostitution and sex tourism. So they have a huge number of people who are HIV positive. Does that make sense? So there's a lot of interesting things about this. Now, this is the most infectious disease that we know, most contagious disease that we know, measles. The example sort of model example of how infectious the measles is. Imagine that you have a corporate party. Say 200 people attend the corporate party. You have room, music plays, people chat. You walk in that room and you are sociopath. You don't like people. You don't talk to anyone. Just stand in a corner for a couple of hours. Then you leave the party. But you gave them the gift, measles, if you had it. It's spread by respiratory routes. It's extremely infectious. That's why, for instance, vaccines, MMR vaccine especially, measles, mumps, rubella, it should be done strictly following the calendar because kids are very prone to measles. And my wife's friend, that I don't really know her well, I know her son died of measles between 2 vaccinations. So he was vaccinated, he didn't have a complete immune response, acquired it. Where? Nobody knows. Maybe some other kid who wasn't vaccinated. Okay. And he died of measles. You couldn't save.
They said measles has been making a resurgence.
Oh, we'll talk about it, yeah. So, yes. Measles? Yes, yes. They had actually the Mickey Mouse with little, it's called, the little little dots. I forgot how they call this rash. Koplik spots. They pictured Mickey with Koplik spots, and he was kind of promoting the idea of vaccine, you know, so you don't look like me and so on and so forth. Yeah, it was an outbreak, and it's regular in Amish communities in Ohio and Pennsylvania because they don't vaccinate. Okay. So it gives you an idea what basic reproduction number represents and how different it is between the different pathogens. Now, before we move on to different other parts of that topic, what you have to know. So remember, first slide we talked about residential, transient, opportunistic. Know what they are. Understand that microbiome is different. Okay? Understand that how babies acquire the microbiome. Right? Now, here, difference between communicable and non-communicable. Recognize them. So I may ask you, which of the following is an example of non-communicable infection? I can explain you what happens if you don't understand, if you don't know that particular infection. I'll take my time and explain it to you during the exam. But you have to recognize it can be possibly transmitted from person to person. Right? More or less contagious. Basic reproduction number, what you have to understand, it characterizes the ability of pathogen to be transmitted. The higher it is, the more people will be infected by the sick person during the course of infection. Are we clear on that? And no, you do not have to know any basic reproduction numbers for the infections. No. We'll not ask you what is the R0 for measles. Forget it. Okay. We good? Nosocomial infection. Acquired in a hospital. We discussed this, why nosocomial infections are common. You have a lot of sick people in the close quarters. That's it. Okay. Another interesting term and that you have to know is iatrogenic. Iatrogenic infection is the result of the medical procedure. So, for example, person gets, I don't know, you need a spelling? Sure. IATROGENIC. Okay, iatrogenic infection, result of a medical procedure. Now, what can be iatrogenic? Can you think of any examples? Because I have some. Huh? Okay, any type of post-surgical infection. Okay.
Like staph, like sometimes common in like knee replacements or
Absolutely. Otherwise, person wouldn't acquire it, right? If not for the surgery. An infection that stems from the transplantation, well, after the transplantation, after the immunosuppression. Any infection that comes up because of the immunosuppression is iatrogenic infection. It can be both, because if the microbe is acquired in a hospital environment, it's both iatrogenic and nosocomial. But you have to appreciate that, for instance, I can say if I, with my child and I would go to the doctor's office and we seek in the part that's for sick kids, you know how they sometimes have separated, and my son is sick and I'm not sick, we come home, I become sick. Maybe it's nosocomial infection. Maybe I acquired it from someone in that hospital environment. Does that make sense? Another word is zoonotic. For some reason, I keep saying zoonotic. Technically, according to English, it's zoonotic. Z-O-O-N-O-T-I-C. Can you think of any zoonotic infections acquired from animals? Mad cow disease, zoonotic. Lyme is zoonotic. Avian flu zoonotic. Anything that comes from animals is zoonotic, right? And oh, West Nile, yeah, absolutely. West Nile zoonotic. I can think of very few diseases that we do not know animal reservoir for. It's a smallpox, which is eradicated completely. Measles, by the way, which, speaking of which, smallpox, how was it eradicated? Anyone knows? Yes, they vaccinated everybody. And by saying everybody, I mean it. Every person on the planet got immunized. So virus had no host to replicate it. We have no smallpox on Earth. Measles is also entirely human infection, 100% human. Can we eradicate it? We can. Well, if not for people, we need dictatorship for like 6 months on the entire Earth, so everybody receives measles vaccination, and then we're good. It's going to be gone, because the close relative of measles, called rinderpest virus, it's a cow infection. It's eliminated in the U.S. completely. They vaccinated all animals. Okay, rubella is gone in the United States because of the vaccination program. It was successful enough. Okay, with measles we didn't succeed that much. So if we look at something that's absolutely human and only human, we can eradicate that. Okay, think about this. Now, pathogenicity and virulence. These two terms create a lot of confusion, not only in students, but in scientists as well. That's why I want to precisely define how I want you to answer my question about this. Pathogenicity is qualitative parameter. It is not measurable. Pathogenicity means whether the microbe can or cannot cause the infection. Do you see what I'm saying? So say influenza is pathogenic. Staph epidermidis is non-pathogenic. Okay? Does that make sense? Now, virulence is how severe the disease can be. Some microbes are considered to have low virulence. Some microbes have high virulence. Can you think of any microbe that causes the disease that's pathogenic but has low virulence? Not really severe disease. Why are you so shy? Come on, say something. Huh? Microbe that's pathogenic but has low virulence can cause the disease, but disease is not really severe. Common cold, perfect example. It's very contagious, pathogenic, but low virulent. Can you think of a microbe, also pathogenic, that has high virulence? Tuberculosis, highly virulent microbe. There's a great example of the microbe that is not communicable, has ultimate virulence. It's rabies. If you get rabies and you don't treat it, you're done. We have 3 cases, I think, of over like span of 100 years. People who were cured from rabies. You're going to die. So that's extremely high virulence. Ebola has high virulence. Different strains of influenza may have different virulence. You probably have heard about influenza A, influenza B, influenza C. Influenza B has lower virulence than influenza A. It usually causes milder disease. Does that make sense? Do you see the difference between pathogenicity and virulence? You have to be able to recognize that difference. Okay? Both pathogenicity and virulence are determined by virulence factors. We have had some examples of virulence factors before. We talked about Griffith experiment. When we talked about bacterial transformation, we had 2 strains of Streptococcus pyogenes, one having capsule and another not having capsule. The one without capsule was not pathogenic, so capsule was the virulence factor. Okay. Now, one thing that we also have to appreciate is that the virulence of the microbe, in some cases, even pathogenicity of the microbe, can be determined and is determined not only by the virulence factors of the microbe, but also by the health immune state. Okay? For instance, there's an infection transmitted by ticks. It's called babesiosis. The causative agent is babesia, and I don't remember the species name. Generally, people develop fever, and it's self-limiting infection, which means no treatment is necessary. Generally, for a healthy adult, babesiosis will recover. A person with babesiosis will recover. However, if the person underwent splenectomy due to, I don't know, lymphoma before that, people with splenectomy are pretty much doomed if they get babesiosis, because T cells, the splenic T cells, are extremely important in eliminating the protozoa. Okay, so you can see one thing completely changes the outcome. In people without the spleen, babesiosis is extremely virulent. Okay. So sort of, it's not only genetic, general level of health. Okay, genetic profile. That's a great example. There was a case few years ago when the girl, without any apparent abnormalities, at the age of 6 months, developed extremely severe influenza infection. She was on the brink of death, and nobody could figure out what's wrong with her. And then doctors actually sequenced the entire genome of the girl, and at that time it was about $10,000, and they found that she had a mutant gene that was responsible, one of the genes responsible for the production of interferon. So they knew what's wrong. They kind of heightened up the interferon treatment. She recovered. Otherwise, she's perfectly healthy. There is absolutely no phenotype when she's not sick. When she's infected with the virus disease, she's getting sick. She's much, much worse than all other kids. Okay? So genetic profile. And of course, previous exposure to microbes plays a role. I mean, if you had, I don't know, West Nile before, you're not going to get the infection again. Okay? It's not true for all of them, but for most of them. Another great example of genetic profile is plague. First, I think I mentioned that. First plague killed, according to different estimations, from half to 1/3 of the population of Europe. Second, which was a few centuries later, took away about 15%. Then there was an epidemic, I think in the 19th century, when we're talking about percentages. Why? Why the number of people that died of plague during different epidemics progressively decreased. It's not only this. Who survived in the first place? Okay, who died? What was the difference between those who died and those who didn't? Not really, not that much. Which is the phenotype which is encoded in DNA. Make sense? So we got selection. Always remember that the species, the people that you see around you, are those who survived. Okay? Unlucky died. So those died, and they kind of read the population of the genes that were not resistant, did not provide resistance to plague. And that happens, you can see it all the time. Okay? And why I'm telling you all this, you have to consider this when you have a patient in your hands. Okay? When older people are immunosuppressed, people who had transplant are immunosuppressed, people, cancer therapy, immunosuppressed. My wife's colleague, he had leukemia, so he had bone marrow transplant, takes immunosuppressive drugs. Well, guess what? If somebody coughs in the lab, he leaves for the rest of the day because close quarters. He doesn't buy lunch. He brings his own. He rarely eats outside because risk. He's immunosuppressed constantly. A very good friend underwent chemo. It was awful because when we had like a little gathering for friends, if somebody was sick, we had this situation. And sick, I mean somebody was like sneezing. Do we call the sneezing person, or do we call a friend on chemo, because they cannot be in the same room together? Okay, so immunosuppression is the great factor. Age is the great factor. For instance, inflammation of the brain is much more dangerous in kids than in adults. In adults than in kids. Why? Why encephalitis, brain edema is much more dangerous in adults than in kids? Exactly. In kids, skull is soft. So even the brain, if the brain is inflamed, skull can stretch a little bit. In adults, it cannot, so people are at the high risk. Okay? Does that make sense? So there are a couple of things that we haven't discussed yet. It's the infectious dose and a portal of entry. So we're going to talk about infectious dose first and then the portal of entry. So infectious dose. Number of microorganisms that are needed to cause the infection. As simple as that. You can see that it varies greatly from one microorganism to another. My favorite virus, well, it's not my favorite, second favorite virus, norovirus. Its infectious dose is under 10 virus particles. That's the same virus that causes two-bucket disease. Okay, it's really little. On the other hand, if you would look at different strains of E. coli, actually, they show you a great example of sort of correlation between the virulence and infectious dose. So enterotoxigenic and enteropathogenic E. coli, different strains. The main manifestation of infection is diarrhea, and that's it. That makes sense? However, if you would look at enterohemorrhagic E. coli, this infection can cause dysentery. What's the difference between dysentery and diarrhea? In dysentery, there is something present in the stool. Dysentery is a symptom. It's like a cough that can be caused by different microbes. So what's the difference, clinical difference between dysentery and diarrhea? Again, it's a clinical symptom. Like cough can be caused by infectious disease or lung cancer, but it's still cough. So dysentery, there's something present in the stool. Blood. Yeah, dysentery is the bloody stool. Diarrhea is not bloody stool. Okay? So EHEC, enterohemorrhagic E. coli, can cause dysentery. Hemolytic uremic syndrome has pretty high mortality rate, and so on and so forth. You can see that it has higher virulence and lower infectious dose. Does that make sense? The idea. The general correlation is that lower infectious dose is associated with the higher virulence. Does that make sense? Now you can see that for some of them, the infectious dose is exactly 1 microbe. And let me tell you something, that's the one that you worked with. That was in the tube. Salmonella typhimurium. That's the one you inoculated the plates with. And as far as I know, we didn't have any incidents so far. Nobody inoculated themselves. Okay. Now, you have to be careful though, and I warn you not to compare different species. Do you understand what I'm saying? So say Vibrio cholerae has an infectious dose of 1 million cells. Salmonella typhimurium has an infectious dose of 1 cell. Does that mean that salmonella is the worst disease? Salmonellosis? Not necessarily. If you ever, nobody had cholera here, but cholera is pretty awful. Okay? You can compare different strains. You can say that this strain of a microbe has higher virulence and therefore lower infectious dose. Or we know that this strain has lower infectious dose, so it must be more virulent. Does that make sense? It's comparing different species like comparing apples and oranges often. Okay. Now, when we talk about infectious dose, we use the parameter that is called median infectious dose. Okay? Median infectious dose, well, that's slightly, so median infectious dose is the one that causes infection in 50% of inoculated animals or inoculated humans. Does that make sense? Only 50%. Because it's fairly easy to establish it using statistical methods. Not 100, not 1, 50%. It's called ID50, infectious dose that causes infection in half of the population. Now, median lethal dose is the number of microorganisms that you need to put into animals or humans to kill 50% of animals or humans. Does that make sense? That is accepted way to compare them. For instance, if you have two different strains of the bacteria, you can say median lethal dose for this bacteria is 10 cells, median lethal dose. Let's play an example. So you have bacteria A. Median lethal dose is 10 cells. Bacteria B. Median lethal dose is 100 cells. Which one is more virulent? A or B? A. Lower lethal dose means that you need less microorganisms to kill someone. Does that make sense? Good. So understand that idea. Again, you don't have to memorize that table. Forget it. Don't memorize this table, but understand first of all, understand what is infectious and lethal dose, and understand the relationship between the infectious dose or lethal dose and virulence. That it's kind of the opposite. Portals of entry. Of course, for communicable diseases. Non-communicable diseases have none. Just examples. Let's just not look at the table. It's for your reference, but let's not look at the table. Let's figure out which portals of entry can you think of and what microbes can use them. Who starts? Who wants to start? Mouth. Which microbe? Salmonella. Perfect. Salmonella gets into the mouth, causes intestinal infection. So we can say mouth and intestines, digestive tract. Let's call it digestive tract. Eyes. Great. Yeah, pink eye. Yes, it can be viral, herpes. It can herpes conjunctivitis. It can be bacterial, like Staph aureus. Great. Ear. Ear infection. Mostly, though, the portal of entry would be respiratory, upper respiratory tract, because it would get from the inside. But you can, I can think of the outside entrance. Okay, it's going to be like Haemophilus influenzae, Strep pyogenes. Something else. So we got upper respiratory, we got GI, we got eye. Skin. Yes. Usually it should be damaged. Some microbes do not have skin to be damaged. Can you think of any? Microbes that can just grow on the normal skin. Yeah, but it doesn't cause the disease. The skin disease. Infectious skin disease. Great. Impetigo, Staph aureus. Great. I thought about fungal infections, but impetigo is perfect. A great example. You don't need to have broken skin. If you get pathogenic strain up, okay. Skin. Holes in the body. So we covered mouth, we covered upper respiratory, nose, ears. What other holes in the body we have? Anus. HIV, really. Let's be honest, I don't know about you, I can hardly imagine conditions of voluntary transmission via anus except during the anal sex. Okay, and that's going to be HIV. Yeah, perfect. Sexually transmitted diseases. And anatomically going in line with that, in men it's just the same tube. UTIs, yes. Same. And this actually brings us to the moment. Acquisition of microbe via unusual portal of entry may lead to the unusual presentation. What I mean by that? Urinary tract infections. Different symptoms, yes. Urinary tract infections. Which microbes cause them? E. coli, by far the most common. Where does it come from? Feces. Which population, which half of the humanity is more prone? Women. There's one microbiologist, that's not my words, that's the words of Dr. Michael Schmidt. Women have internal anatomical problems with plumbing. Anatomically, women are more prone to urinary tract infections. E. coli in the intestines pretty much doesn't cause anything. E. coli in the urinary tract causes massive inflammation. Okay, so you put microbe in, then you get it through the unusual place, and presentation can be completely different. One of the examples that I often give students, yellow fever is the disease that is transmitted by mosquitoes. If I would have a can of cell culture fluid with yellow fever virus, it would taste lousy, but I would drink it and nothing would happen to me, because the only portal of entry that allows it to establish the infection is via the blood. Needle, mosquito, doesn't matter, transfusion, whatever. I drink it, nothing happens to me. It cannot infect via the gastrointestinal tract. I can bath my hand in it, as long as I don't have any cuts, I'm fine. Does that make sense? So portal of entry is very important for establishing infection. Now, one portal of entry that we never mentioned, in the left bottom corner, torch. Placenta, vertical transmission. It is an important route for congenital infections, infections that children are born with. So TORCH is the acronym that stands for Toxoplasma. Other, I really like other moment, it's very creative, which includes a whole bunch of infections like syphilis and hepatitis B and chickenpox and hepatitis C, HIV, parvovirus B19. Then R stands for rubella, CMV, and herpes simplex. All those infections can be transmitted vertically. All those infections can cause serious problems for a child. Extremely serious problems. Now we're going to talk about attachment. We're not going to talk about invasion. So stages of infection. Microbe first has to attach to the surface, right? Or adhere to the surface. How a microbe can attach to the surface? Bacteria use different types of proteins called adhesins. They use fimbriae. They sometimes use pili, capsule, slime layer. So think about this. Any external appendages that make microbes more sticky, that bacillus species that we saw on the plates that had capsule, it makes it more sticky. It attaches to the surfaces better. Okay, appendages like fimbriae and pili will increase. Often fimbriae are composed of the proteins called adhesins. Okay, now you have to appreciate that so those are all virulence factors. If a bacteria has genes for adhesins, it means it can better stick to the surface of the cells. Does that make sense to you? So it will remain in a place longer. You often can just, if you take away the gene for adhesins, microbe may be not pathogenic anymore. Does that make sense to you? Right? Good. Now viruses. Now let's talk about viruses next time.